Hello, welcome to class. My name is Annika Pratt. I am a junior here at Concordia, majoring in biology, computer science, and Spanish. And this is my research project for the semester, Identification of Elodea Species by Differential Restriction Digest. So Elodea is an aquatic plant that is native to the United States and invasive in Europe. If you look over at my little box behind me, that's Elodea. The two most common species are Elodea nutalii and Elodea canadensis. In the United States, these species can be distinguished just by looking at them, but in Europe, where they're invasive, they are developing similar morphology, so it's harder to distinguish them just by looking at them. A few years ago, Concordia students sequenced the internal transcribed spacer regions of the ribosomal DNA of both species from both United States samples as well as French samples. In these sequences, they found a 14-point mutation difference between nutalii and canadensis. The problem with sequencing is that it is expensive and a hassle because it can't be done at our own lab at Concordia. We have to send samples to a different lab to be sequenced. So we decided to try using a restriction digest to distinguish between these two species in our own lab. Restriction digests involve restriction enzymes, which cleave DNA sequences at specific locations. The restriction site for the DPN2 endonuclease is GATC. This GATC sequence appears 3 times in the ITS region of Elodea nutalii and only once in the ITS region of Elodea canadensis. So 2 of those GATCs in nutalii are at those point mutations between the 2 species. So I used bioinformatic tools to see where the DPN2 enzyme would cut each species and what DNA segments would remain after cutting. Based on this analysis, I hypothesized that a DPN2 restriction digest would yield 2 bands for each species, with the pair of canadensis bands being slightly larger than the nutalii. So the PCR products of the ITS region of both species is about 700 base pairs in length. Those GATC segments in nutalii, there's one about 15 base pairs in, and then about 223 bases after that, there's another one, and then there's 11 bases before the next one, and then you end up with about 440 bases left over. So when DPN2 cleaves the ITS region of nutalii, you end up with 2 really small segments of DNA, and then a segment of DNA that's about 223 base pairs long, and one that's 440 base pairs long. Canadensis, on the other hand, only cleaves once, and that's about 235 base pairs in from the end of that segment of DNA. So you end up with one piece of DNA that's 235 base pairs long, and then another segment of DNA that's 454 base pairs long. So using gel electrophoresis, we wouldn't be able to see those really small bands from the nutalii. That's why I hypothesized that we would see 2 bands for each species, but because we cut off those little segments from the nutalii, the canadensis bands would both be larger than the nutalii. So for methods, I used PCR samples that were created by Concordia students back in 2019. I had those samples digested with the DPN2 enzyme for about 15 minutes in a 37-degree water bath. Then the enzyme was inactivated in a 65-degree water bath for 20 minutes. Each trial consisted of a negative control, so there was no enzyme, and then also the digested. So I've run 7 trials of this experiment so far, consisting of 6 different samples of each species. These trials used an equal number of Nutalii samples from the United States and France. So I used 3 Nutalii samples from the US and 3 from lakes in France. Canadensis trials, I used 4 samples from France and 2 samples from the United States. And I didn't see a huge difference between the French and the US samples, which there shouldn't be because GATC is not within any mutations between the French and the US, and there's only 2 mutations between those 2. So let's look at some results. This gel, figure 1, shows the results I saw most frequently of all of my trials. So 4 of the 7 trials looked like this. In lane 1, we have a high-low marker. So this is consists of DNA fragments of known sizes. So the larger ones are on the left, the smaller ones are on the right. Lane 2 is the PCR product of Nutalii. So this is my negative control. It's about 700 base pairs, just like I would expect. Lane 4 is the negative control of Canadensis. This one is a little bit larger than I expected, and I saw that frequently, that Canadensis was larger than Nutalii. I'm still trying to figure out why that's happening, but it is. Lane 5 is the Canadensis digest. So there's 2 bands like we expected. This larger band is a little bit larger than expected, but that's probably just because the PCR product was larger than expected. The Nutalii digest is here in lane 3. This is not really what I was expecting. So I was expecting one band to be a little bit bigger, and these 2 bands are both around 250, 300 base pairs. So they are smaller than the 223 and the 440 that I was expecting. One of them is right on, and the other one's a little bit smaller than I was expecting. Unfortunately, there were 2 Canadensis samples that did this same thing. I'm not sure why, probably due to some human error, but I am looking into that. And then there were 3 Nutalii trials and 1 Canadensis that didn't yield any results. Either both the negative control and the digest didn't have any bands, or just the digest didn't have any bands. So continuing on with this research, I'm going to continue trials, because if I continue to see results like this figure 1, this could actually be a good way to distinguish between Canadensis and Nutalii. This would actually be an easier way to distinguish between them than what I had originally hypothesized, because the Nutalii bands are all 300 or less, and the Canadensis has that larger band around 400 or 500. So that could be really good if we continue to see results like this. But if Canadensis shows more banding patterns like the Nutalii, then it won't be a good option. And we can look into other restriction enzymes that cleave in those mutated spots. So I'm going to use bioinformatic tools to try and determine why DPN2 is cutting the Nutalii into smaller pieces than I had originally hypothesized. And then I'm also going to look into why these Canadensis bands are usually larger than the Nutalii. I would like to thank Dr. Pratt and Dr. Marco of the Concordia Biology Department, also Erska, and then other students, Caitlin, Alyssa, and Brooke, for all of their work in this research. It's been a super good experience. It's a really good project. So thank you for listening, and enjoy COS.